All right, we are on chapter 10, section 2, and we're talking protein formation. So this will be a lot shorter than the last one, which is nice, right? So this is actually more of a review because we're really talking about condensation reactions when we talk about forming proteins. Because we take two amino acids and we have a condensation reaction that takes two amino acids and turns it into one dipeptide, right? So what happens when you do a condensation reaction? If you're going to have condensation, this means that we are going to form water, right? So you lose a hydrogen, you lose a hydroxide, and you make one new product, right? So it's the carboxylate ion of one amino acid and the protonated amine from another amino acid that really react together. And that water molecule is removed, and then you have an amide functional group that's formed from that. So what's an amide functional group? Well, let's look at what happens, right? So you have any kind of carboxylic acid or carboxylate ion, right? So in our case, we're talking about a carboxylate ion. So we have that C double bonded to an oxygen, and we have another oxygen that has a negative charge. So now you're going to react that with an amide, a protonated amine group. So what do we need to form water? We need H2O. We need two hydrogens and we need one oxygen. So here's our one oxygen, and here are our two hydrogens. So those are the three things that are going to come off and are going to go and form water. And so what happens when you end up with that, when you finish that condensation reaction, is that this carbon right here is going to form a bond with this nitrogen right there. So you're going to lose that oxygen, and you're going to have a bond form between the carbon and the nitrogen. And so that's exactly what you have here. That's the carbon bonded to the nitrogen. So now we went from a carboxylate ion reacting with a protonated amine to make an amide group. So remember that amide group is our nitrogen bonded to a carbon that is double bonded to an oxygen. And that's really a peptide bond. So you've actually kind of done this before. We've talked about it before when we talked about forming fatty acids and glycerol and that kind of stuff. But really what we want to focus in on now is really the peptide bond formation, right? So here we go. So the other kind of reaction that you can have is you can have a condensation reaction when we're making sugars, right? So remember when we had those ring sugars and you had two sugars, and in order to hook them together, you had a condensation reaction. It's the same thing. And you form that glycosidic bond. It's the same condensation reaction. You're just starting with different materials, right? So here's our example where we have two different amino acids. We have, in this case, an alanine, and we're going to look at the carboxylate right there. And we're going to look at the protonated amine. And those are the two side groups that are going to react together in this condensation reaction. And we're going to get rid of this oxygen, and we're going to get rid of two hydrogens. Those are the parts that are going to go and make water. And then you're going to form a bond between the carbon and the nitrogen. And that's how you get a peptide bond. So now we have a dipeptide, right? It's a peptide, but it only has two amino acids, so we call it a dipeptide. And in this case, whenever we write them, we're going to always write the three-letter abbreviations with a dash in between the three-letter abbreviations, saying that it's participating in a bond. Okay, so in our dipeptide, the N-terminus, right, has an unreacted alpha amino group. So this is our unreacted amino group, right? And then in the C-terminus, or the carboxy terminus, we have an unreacted carboxylate ion right there, right? So we have our N-terminus and we have our C-terminus. So just kind of keep that in mind when we're looking at those structures. So we always write our peptides from the N-terminus to the C-terminus, right? So you need to be able to circle amino acids pick the one that's at the N terminus, be able to name it, be able to pick the one at the C terminus, be able to name it, locate peptide bonds, give your 3-letter and 1-letter abbreviations, all that kind of stuff. So you have to be able to do all of that, and we'll do some practice. So dipeptides can be formed between any 2 amino acids, and each pair will combine to form 2 different dipeptides, because you could have alanine as the N terminus and valine as the C terminus, or you could flip-flop them and you could say valine is the N terminus and alanine is the C terminus. And they're different. They behave different. They have different properties. So each of these 2 dipeptides are structural isomers. They're the same chemical formula, but structurally they're not the same. They're different compounds. And so if you rearrange the order of amino acids inside of a protein, you're going to change its function because you changed its structure. So it's very, very important that things, that when you write them in the right order so that we're signifying the correct proteins. Otherwise, you're actually writing out a different protein. So any compound that contains amino acids that are joined together by a peptide bond, we call a peptide, right? Dipeptides are the smallest ones, but then you could have a tri, you could have a tetra, so on, so on, so on. But as that number gets larger and larger, remember we said proteins could have thousands of amino acids, then we just call it a polypeptide. And then if this polypeptide has biological function, so biologically active, this means it has a function in the cell or body, something like that, right? And it contains 50 or more amino acids, we call it a protein. Otherwise, we just call it a peptide. So a lot of times, there are some biologically relevant things that are occurring in our society, right? So a lot of people you've heard of have celiac disease. So celiac is an intolerance to gluten. And gluten is really a mixture of proteins that are found in grains. And the grain that's most popular in our diet is wheat. So if you eat wheat and you have a gluten intolerance, then you're going to have gastrointestinal reaction to that gluten. So the 2 main proteins inside of gluten are glutenin and gliadin. And so these 2 proteins have regions that are really, really rich in proline and glutamine. And not a whole lot of proteins have this richness in proline and glutamine. And so the body, when this gluten enters the digestive system, some of the peptides are broken down by proteases, and that's great, but it leaves behind these proline and glutamine-rich peptides in the digestive system, and they aren't broken up. And so sometimes these proline glutamine-rich peptides, the glutamine inside of them are modified to glutamate by an enzyme called glutaminase. And it changes that side chain in the glutamine from an amide into a carboxylic group. And so what happens is that change, right? When you convert to glutamine to glutamate, we changed a polar neutral residue into a polar acidic residue, right? Now we have a negative charge over here. This is a charge of -1. So when you add that negative charge to this polypeptide sequence, right, now you have a bunch of them, it causes people to have the autoimmune disorder celiac disease because the immune system sees these peptides and goes, whoa, 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 we don't have those peptides normally inside the body. That must be foreign. And so their bodies mount an immune response to the peptides. And so you have inflammation of the small intestine. You have, because of that inflammation in the intestine, now the intestines don't absorb nutrients the way that they're supposed to, and the person can suffer from malnutrition. So it's very serious. And so if you have a gluten sensitivity, you really should avoid gluten in their diet. And you'll see lots of foods out there that say gluten-free and all that kind of stuff. All right, so let's go do a few quick problems. All right, so here are our section 2, problems, but I put this in this table 10.3 just to remind you of what those common condensation reactions are. There were 3 of them. But what we're really working on, right, is this one that's the peptide bond formation. So this is kind of the most important one. So if you look, here's our first one. So write the products for the following: condensation or hydrolysis. Remember that the opposite of condensation is make water, hydrolysis is break water. So one is going forward in the reaction and one is going in the reverse. So just kind of remember that. So in this case, what I'm doing here, I look at this and I go, you should automatically recognize I have an amino acid, right? NH3, and I have a C double bonded to an O, and I have a carbon, right? So I'm going to have a condensation reaction here. So we're going to say condensation. And remember, what's going to have to happen in a condensation reaction is you need 1 oxygen and you need 2 hydrogens. So really, what is this H3? Let's just get rid of that and let's put a hydrogen and a hydrogen, right? So what are you going to need? You're going to need 1, 2 hydrogens. So what are you going to make? Well, I'm going to make exactly the same thing. So let me draw in black, I guess. I'm going to draw exactly what I had before. So I have an NH3, that's a positive, C, CH2, OH, hydrogen, C double bonded to an oxygen. Now, I have that double bond, but this oxygen has got to go. So if that oxygen's got to go, this carbon is going to go in and make a bond with that nitrogen. Well, that's interesting. Okay. So now I'm going to make a bond with that nitrogen. Now, that nitrogen still has 1 hydrogen attached right there. So I'm going to go ahead and put it, and then I've got to connect it to the rest of the stuff. So now just go in and rewrite the rest. And that's it. And then really you are making water. But I think a lot of times in the homework, they don't make you write the water. They just say the main product, right? And so we don't consider water the main product. Okay, so in our next one, it's kind of that. We're looking at it, okay, now I have something, how do I go in reverse to a condensation? So now I'm actually going to have to break water in order to break up this big substance, right? So remember, a lot of times what you have is R bonded to an oxygen, bonded to another R, and then we're gonna use water to break that up. So remember, water is HOH, so that oxygen is gonna pick up one of those hydrogens, and then the R group is gonna get the OH, right? So that's exactly what's gonna happen. I'm gonna take a hydrogen and I'll attach it here. And then instead of that carbon, now that carbon is gonna be bonded to an OH. So then just kind of rewrite what you get. We're gonna say CH3 double bonded to an O. Now, instead of that oxygen being bonded to the next carbon, we're just gonna put an H. And then here, we're gonna start with our OH and we're gonna bond that to our CH2, and we're gonna have 7 of them just in a row, and then CH3. And sometimes you'll see it in a different way. Sometimes you'll see it just because it kind of makes a little bit more sense like that, but it doesn't matter. Either way, it's correct. Okay, that one we're doing in class. Here we go. Consider the following tripeptide. Okay, we're gonna have to answer a bunch of questions about it. Circle the N-terminal amino acid. N-terminal is the one that has the nitrogen on the end. So remember, every amino acid only has 1 nitrogen in the backbone. So as you go across, you start at N to get to the end of the amino acid, stop before the next N. So it's gonna be a big circle. So I stop before the next N. So you see how I have 1 nitrogen, my backbone goes nitrogen, carbon, carbon, and then the next nitrogen is in the next amino acid. So don't circle it, right? Okay, so circle the N-terminal amino acid and give its name. Look at that structure, you should go look it up. So the N-terminal is glutamine, and that is GLN and Q. And you can always pause the video and go and look at your sheet and then press play so that you're not just copying down what I write. You need to learn to use the sheet and not just copy what I'm doing. Okay, draw a square around the C-terminal amino acid and give it a name. Now remember that you only have one nitrogen per amino acid. So pick up the last nitrogen in the sequence and everything after it is the last amino acid. So this is the C terminus or C terminal, and this one is threonine, T-H-R-E-O-N-I-N-E, T-H-R and T, okay? So give the 1-letter and 3-letter abbreviations for this tripeptide. Please pause it and try. Okay, I'm assuming you've tried. So 1-letter and 3-letter, if you tried and you gave me this, it's not right. What did you forget? You forgot histidine in the middle, right? So what you should have done is to go in and look at this guy, this amino acid right here is histidine. And histidine is his and H, right? So you could, you would give it QHT as the 3-letter, well, the 1-letter abbreviation, which is only 3 letters. Then you would do your 3-letter abbreviation. Your 3-letter would be GLN-HIS-THR. And that's just showing that they're bonded. All right, don't let me trip you up. Okay, draw the structural formula for each of the following peptides. We're gonna do 17 and 19 together. So we're going to draw them and then we're going to identify the N and C terminus for each one, right? So how do you do this? Well, what I know is, see, oh no, I want to go up and get, let me go up and get a peptide backbone from the last video, right? It was this one. So we're going to go ahead and steal this, right? Okay. See if I can pick up where I left off. Right here, right? Yeah, okay. So I know I need phenylalanine and I need glutamate. So what I'm gonna do is I'm gonna put in my basic structure and I'm gonna draw the individual amino acids and then I'm gonna show you how to make that peptide bond, okay? So here's my basic structure and then I know I have an R group and in phenylalanine, my R group is CH2 and then I have a ring right there, okay? So that's my phenylalanine. And then I need to add to that phenylalanine, paste, a glutamate. So what is glutamate? Glutamate is CH2, CH2, and then another carbon, and then I have a double bond oxygen in H2. Okay, so what do I need to do in order to make the peptide bond? Because I need to make the peptide. So what I'm going to do is I'm going to lose an oxygen and I'm going to lose, so 1 oxygen, 2 hydrogens. And I'm going to make water, right? Because we said it's a condensation reaction. So if I go in and do that, oh, so let me do this. I'm just going to copy this whole thing. Paste. Gosh, I love when I can do that. Okay, so I'm going to erase this. So what am I going to lose? Well, I'm going to lose my oxygen. I'm going to lose my 2 of my hydrogens. And then I'm just going to take that one and move it down. So now we have a bond here. And we're going to, whoa, it's the second time I've done that. Okay, and then we're going to move this guy in. And that's it. We've made our polypeptide. Haha, okay, so now identify the N and C terminus. So here is your N terminus, and here is your C terminus, okay? And that's it, it's pretty easy. Oh yeah, I was gonna just draw in an extra piece of paper because Lord knows I'm not gonna be able to squeeze all that in there. All right, let's see. Oh, I think I put it in the wrong place. Okay, try that again. Right, all right, so now my next one, B, is KCG. So B, I need KCG. All right, so I'm going to paste my original. Well, I guess I'm going to paste this one because it was right there. So I'm going to do this. Let's just do this. Okay, and then I need to remember to copy this blank so that we can keep doing this. Okay, so I'm going to have 3 amino acids in this case, right? So what I'm going to do is I'm going to put this in 3 times. 1, 2, hopefully you fit. Yes, just barely. Okay, so I know that for every one of these, I'm going to have to remove the oxygen, but I only remove the oxygens where I'm making the peptide bonds. So if I have 3 amino acids, I only need to make 2 peptide bonds. So I remove my oxygen. I'm going to remove 2 of my hydrogens for each of these, and I'm going to move this hydrogen down, and put in my bond for my hydrogen. And then all I have to do is scooch it all in together. There's my nitrogen, and now I have 3 peptide bonds. All I have to do now is go in and do my amino acid sequence, right? So KCG, K is lysine, or lyse, right? Then I have cysteine, C-Y-S-T-I-N-E, cysteine. Then I have glycine, right? C-I-S-G-L-Y, okay. All right, so now what I need to do is go and look up those side chains. So go in and look up those side chains and then come back. So where am I going to attach those R groups? To my alpha carbons. Those are my alpha carbons. Alpha carbons are those chiral carbons. So for lysine, I have 4 CH2 groups. So I'm just going to do this just so I can squeeze it in. And then I have NH3, and that nitrogen's carrying a positive charge, the way it fits. So cysteine, I have CH2, and then I have SH. And then on my last one, glycine, is only a hydrogen. So that was kind of nice and easy. All right, so the next one, what do I have? His, met, let me see this. C is his, met, and glen. So what is his, met, and glen? That's HMQ. It's my 3-letter abbreviation, HMQ, but it's histidine, methionine, and glutamine. U-T-A-M-I-N-E, glutamine. I think that's right. Okay, so what I'm going to do is I'm going to come up here and I'm going to steal my 3-peptide, right? Copy, because it's the exact same thing. We got 3 peptides. So we have 3 peptides, and then all we have to do is put in our R groups, right? So let's see, I might have to shift that down a little bit. Okay, so the first R group that we put in is histidine. Histidine has a CH2. Then it has that house, right? So then it kind of does this, and we have a nitrogen here and a nitrogen here. And this nitrogen is positive because it's participating in a double bond here, and we have a double bond there. Then what do we have? Methionine. Methionine. Ooh. I think I put that one on the wrong one. There you go. Make sure you get those alpha carbons right. So then I have a CH2, a sulfur, and then a CH3. So that's methionine. All right, and last I have glutamine. Glutamine is CH2, CH2, and then I have another carbon double bonded to an oxygen, NH2. Okay. Let's see, D. What are we doing on D? Where's my D? W and Y. Why are we doing this? So remember, this is tryptophan, right? It trips you up. Think of Elmer Fudd. Tryptophan, right? And the Y is tyrosine. That's the one that gets people all the time. So tryptophan and tyrosine. Right? Oh, and I think we didn't label the N terminus and C terminus on like any, we did on some of these, right? But here's the N terminus. So you're always going to start with your first one, N terminus. So your N terminus is your lysine, And then your C terminus is your glycine, right? And then here, this is my N terminus. So my N terminus in this case is histidine, and my C terminus in this case is glutamine, right? Okay, so now I'm gonna put in my peptide, but I actually only need 2. So what I'm gonna do is I'm gonna remove all this stuff, but then I have to remember, put it in black, that I have an oxygen with a negative there, right? And so now I just have to worry about my R groups. And so the carbon that has that oxygen, that has that hydrogen, not the oxygen, is my R group. So my tryptophan trips me up to draw too. It's kind of big to draw, but it's that house, right? So it's your CH2, and then you have the house, right? And the house nitrogen here with hydrogen, and then the side of the house is attached to a benzene ring. Okay, so that is tryptophan, kind of trippy to draw. Oh, and you got a double bond there. Don't forget to double bond. And then tyrosine. Tyrosine is a CH2 and then goes straight into a benzene ring. And then at the top of that benzene ring, we have an OH. Okay, so that is tyrosine. Okay, and then N-terminus is your tryptophan, and then your C-terminus is your tyrosine. Okay. All right, let's see what else we got. That's going to be for class, and then that's it. All right, y'all have a good rest of your day.